Okay. Good afternoon, everyone, and welcome to the panel discussion. So today, the topic of this panel discussion is population neuroscience. So I know this is the last section in today, so I hope everyone is still excited and not be tired for the whole day talks. Yeah. And so before we start these questions, so I just pull out all these panelists here. So without guest permission with the panelists.
Have to reorder.
Yeah. I just put all these pictures in order from this our program book. Yeah.
How did we know that?
Okay, so just a quick introduction. Yeah, for all the panelists. So today we have Tom Nichols from the University of Oxford, and Shen from University of Pennsylvania, and Dr. Tudorascu from the University of Pittsburgh, and also Wesley and Thompson from the Laureate Institute for Brain Research. Okay, so in next, I think the whole schedule for this panel discussion is we will just give some background of this population neuroscience, and after that, so we will focus on a couple of key topics related to this population neuroscience, and also we pose some questions for each of this topic, and then we have all this Q&A time, and that's the whole picture for this panel discussion. So actually, when I prepare for all these questions, so I just want to get a big picture of this population neuroscience. So I just Google it and want to find this official definition of this population neuroscience. But unfortunately, actually, I cannot find it either from some special official website or from this wiki. So what I can do is I just go to the ChatGPT and hope it can give us some surprise. But actually, so they give a different version of this definition for the population neuroscience. I just pick one of them, which is, I think is maybe it's more accurate for this topic. But if you have any comments, yeah, you're very welcome. And so for the population neuroscience, actually is an interdisciplinary field that explores this relationship between the brain structures, functions, and behavior across large and diverse population. So it also integrates different methods from the neuroscience, epidemiology, genetics, and also data science. And all these things will be used to study how these different factors affect our brain health and also cognitive functions at a population level, okay? So there are several of these key focus areas, so including like the large-scale studies and multimodal data sets, and neuroimaging and brain function, and genetics in environments, lifespan, perspective, and public health implications. And also it can be applied to different domains. For example, the epidemiology of neurological disorders and risk factor identification and some targeted interventions. So in this discussion panel, so we are focused on two different aspects. So one is the large scale study and another one is the new technology. So let me just give some background for these two different topics. So actually, as we know, in the past 10 years, so from this government and different countries, there are a lot of this kind of large scale studies launched. And you can see that, for example, in the UK Biobank, the HCP, ABCD, and ADNI. So all these studies and data set, I think, has been mentioned in these talks from yesterday to today. And so you can see that if you take a look at these numbers in each different studies, the number of participants has increased exponentially. And you can see, as I remember when I first jumped into this field, and typically, if you can get a data set with a sample size around 100, 200, that's a really big data set. But this 10 years or 15 years later, today, if we just if we just look back to all these studies, so all these previous studies, like small sample studies, because you can see like UK Biobank, now the sample size is around 500,000, so that's a lot. So that is about the large scale studies. Actually, we also have all the things that is for the data sets. So instead of you only have one multimodal data set like the structural MRI or the T1 weighted image. So today we can connect this kind of data set from different levels. For example, we can connect not only this kind of connectome based data, functional connectome, or this structure connectome data. We also can connect this neural level data. We can also connect this kind of behavior and the neuroscience, neurological level data. So all this data will be integrated together and for us to investigate some scientific questions. So besides this data sets, we also interested in some new technologies. And everyone knows that. So today, with the highly development of these AI-based methods, so all these technologies has been involved into these population neuroscience studies. For example, when we get all these data sets, we can use these AI-based methods and to help us to do something like this kind of this early prevention and this the classification and disease disease diagnosis, and also it can help us to refine this disease treatment. So I think now we will just go to this Q&A part. So for the first question that is related to, actually this is not only one question, we have a sequence of questions here. So the first part is about the large scale studies. So the question is, so what do you see as opportunities and challenges in conducting the large scale studies? And what role do our statisticians and data scientists play in addressing these opportunities and challenges? So I think we just start from Wesley.
Well, yeah, okay. So you can call me Wes. So I can just talk about like my experiences as part of the ABCD study, and we're about 10 years in, and we're already planning the next iteration of ABCD. So it's which will happen in 2 years, and so we've been thinking a lot about like what lessons we've learned from the study and like how we'd like to morph it going forward. One thing, I took some notes. I'm not gonna go through all of them right now, but like just a big one for me is like, so population neuroscience, to me, it means like a large data set that's reflective of the socio-demographic diversity of the population of interest, which in our case is the United States. So that addresses a key point of bias, which is external validity, which is that like your results that you have are relevant for the population that you're interested in applying it to. And the United States is a very socio-demographically diverse country. So you need a large sample in some ways to be able to reflect that diversity. And this is really this is really about epidemiology now. And so there's not a lot of like, there's a lot of very sophisticated like data analysts who work in neuroimaging, but there's not a lot of neuroimagers that have epidemiological expertise or think epidemiologically. And so they'll say, well, we need large studies that are diverse to reduce bias. And they don't define what they mean by bias. And what they really mean by bias is external validity, the lack of, that your results reflect the population of the United States in this case. But they don't then think about, well, if you have a very diverse sample where a lot of the effects are small and diffuse, and sociodemographics actually is an order of magnitude higher association with a lot of outcomes, internal validity is also an issue, and that's just that your results are not being biased by confounding factors. And so how this increase in diversity actually makes it perhaps more likely, maybe we can debate that you're going to find results that are confounding. And so I guess like one lesson that I've learned, and one thing that I think is really relevant, is really bringing this whole epidemiological literature about like different sources of bias. It's not just internal, external validity, it's also measurement error bias and selection bias and other types of bias, and applying that to these large data sets. I have some other notes, but I'll hold them off.
So this goes back to how can we make an actual impact, right, as statisticians. So I think the opportunities here limitless, right? We can develop methods. We can be directors of the data cores, which are part of these large trials. And I think that's where we can really make a difference, from data collection, from data integration. I think I learned a lot from the multi-site studies, specifically with respect to standardization across all these, lifestyle questionnaires that we administer, cognitive measurements that were administered across different centers, how we make mistakes in the beginning, how we measure them on different scales in one center, different scale in another center, and then putting them together. So I think there are many opportunities in terms of, methodology, and as well as putting our, skills into practice here.
All right, so my perspective about this large data collection is certainly it provides a lot of new opportunities, right? First, statistically, it's more powerful, right? So you can detect a lot of signals that can be detected in smaller data set, right? In terms of machine learning, and that can address the risk for overfitting. You also need a lot of data, right, to train the model. So as long as what we can do, people actually have already been doing that, right? So neural network has been there for a long time, but now it became popular kind of recently, right? Because there's a large data, right? Another thing is we have computational kind of infrastructure is much more powerful. That's why it actually make a big impact, right? So I think that's the power of big data. And another thing is now, with generative AI, now we can build these foundation model, right? So, and this is also because of the kind of the large data, right? Now you have a humongous data that you can train some foundation model, and also that can be fine-tuned towards different applications. So you can also address the disparity, right? So like a segment anything. So you have all these different images, right? But now we have this very powerful tool that can build a foundation, and then it actually works on a lot of existing images, but for specific images in the medical domain, right? Say like MRI scans, radiology report, then you probably need your local data to fine-tune those. Those are very powerful tools, actually. And another thing is, since the data is humongous, actually you can study some more complicated problem, right? So in terms of, since I studied dementia, then it kind of Alzheimer's is not a single disorder, right? You need to look for different subtypes. Then you can find the underlying mechanism, identify target, look for drugs, right? If you mix those together, you won't be able to find those drugs, right? So in that case, you can actually look at the subtypes, right? Data-driven subtypes. And at the same time, people also want to model the disease progression, right? So you can actually model those together, right? Look at both subtypes and staging, right? So there are existing tools that can help you to do that. I think these are something actually we can work on. So look for hopefully more complicated and interesting patterns. And certainly these need to be driven by the actual problem, right? So I think now we are more powerful in terms of kind of define those more meaningful biomedical problems. Yeah.
I would also echo something that Wes said about epidemiology, because I think that's very easy to just say, well, I'm going to find something in there. And, if you do that, you'll find that, yogurt consumption, shrinks, the frontal cortex or something. So yeah, that epidemiological thinking about sort of coming in with a question, of course, that then depends working closely with collaborators, what is the interesting question here? In terms of what can we do, what can we play, I guess the thing has been I've been talking with a lot of people here about the changes coming with the UK Biobank and how they're not going to let people download the data anymore. I'm hoping to extend that, delay that as long as possible. But I think for a lot of data sets, that's unfortunately the future, that they're going to be on lockdown, in cloud resources and what we call trusted research environments. Some people say secure research environments, because what does trust mean? But anyway, SEEs, TREs, what have you. they are going to be the future. And so I guess the thing I would say is that it's kind of right now they're awful. All the ones I know about are really bad. So I think something that statisticians can do is to work with the people that build these to figure out like why are they so good? What can we do to change these? Are there standard suites of tools? What are the things that are actually really sort of in making it difficult to work with? And I've started that discussion and definitely fed that information back to the UK Biobank and the Research Access Platform run by DNA Nexus, which you can tell from the name was oriented around genetics and not imaging. And I guess what I can tell you from that is that there's 2 years left on that contract, and they're then going to go out for bid again on that. So they are looking for input on what should they be looking for, and I'm sort of collecting ideas and reporting them back. But that's where we can. That's one of the ways we can put in feedback information.
Thank you. So I think let's move to next one. So we know that when we got all this data set, actually there is a key part that is how we can process this data set, and also we can integrate all the data set from different studies or from different sites, and then we can just empower this kind of data set and apply it to some specific scientific questions. So the second question is about that. So how can we enhance the current pipelines for processing and integration of these data sets, especially these large-scale multimodal data sets? And also, what contribution can we make to this neuroscience community or the public health by using these resources? Okay, Wes.
I'm going to interpret this question broadly, otherwise I'll have absolutely nothing to say. So I'm going to talk about integrating imaging data into a broader study that's collecting lots of different types of data, if that's okay. So I'll leave the pipeline questions to other people. So one way I think about like ABCD and other large epidemiologically focused studies is how why are we collecting brain imaging? Like, why is ABCD collecting brain imaging? It's an NIH funded study of public health. Like, what does brain imaging tell us about public health? It's a large, expensive part of the study. It took resources away from other things that they could have measured. You can't intervene directly on somebody's brain. There's no health intervention that I know of that intervenes directly on your brain to make you better or worse. And so why are we collecting this information? And so I think a key way of thinking about it is the brain is the mediator. And so if you're studying behavioral outcomes like psychiatric outcomes or substance use behaviors or any type of cognitive behavior, you have to study the brain because that's where the behavior resides. And so if you're looking at the effect of some exposure on a behavior, it's natural then to think of the brain as a mediator of that exposure and to understand so one thing that I like to do is encourage scientists naturally think about this. They don't necessarily put it in this framework formally, but like you have an exposure, it could be genetic, it could be environmental. You have some outcome of interest. It could be like depression or some other mental health variable, and you want to see how that exposure is instantiated mechanistically, biologically by looking at its impact on the brain. And so I think, and then how to incorporate multimodal imaging data into that mediational framework, especially if it's collected longitudinally. I think that's something that we need to develop novel methods for. I know Martin's done some work in this area, but I think we need to come up with better ways for applied scientists to be able to use the brain imaging data as a mediator, because it's not very good as a predictor of outcomes. You can get a family history a lot cheaper. That's a lot more predictive. So
I think one of the questions is, do we really want to integrate all this data? I think, you can answer both ways, yes and no. The integration of this data that we have been collecting comes with a lot of challenges. Pipelines, from my perspective, we've analyzed data to different centers, same pipelines, and sometimes we get 40% differences in measurements that are very small from the brain, right? From MRI, cortical thickness, or other measurements. And that's because there is always some sort of a parameter that is not fixed correctly. So it's very challenging. I think transparency is one thing, to put out your code, to collaborate with a very great team of people that have different expertise, that can answer some questions that you wouldn't know as a statistician. And then I would say that there are some other challenges that come with contributions that we can make to the neuroscience community and public health using these. I think we should always start with a question, right? Going back to the epidemiology, what is the actual purpose of combining these data and analyzing them together? Is there, besides having a huge sample size or trying to look at trajectories of disease or having more power or things like that, what is it that we cannot get from analyzing studies separately? So we have to put them all together. So different ways to look at the problem, I guess.
Can you hear me? Oh, okay. So yeah, actually, I completely agree, and I think so in some cases we may want to integrate those together, right? So if you want to say increase the statistical power or reduce the risk for overfitting, right? So but in that case, we have to kind of like do data harmonization, right? Because a lot of these data are multi-center kind of data are collected from different institutions and medical centers, and to put them together, we have to harmonize those, right? So there's a lot of statistical machine learning methods that can help to do that. So I think we have to be careful so when we integrate all those data together, right? And but there are different strategies, right? So you can harmonize those and put them together, then you can do some mega-analysis, but you can also do some meta-analysis like Paul Thompson, Enigma, right? So, and it's also huge, right? But you just collect the summary statistics and then do meta-analysis on that. So you can also detect some signals, which is kind of missed by these individual studies, right? So, or if you're concerned about the say privacy security issue, then there's a federated learning framework, right? You can do like to integrate all these kind of data together. So that's more like integration. And in terms of analysis, I feel like maybe I probably from the multimodal perspective, because we collect all these data, then you have multimodal imaging, structural, functional, diffusion, perfusion, MRI, and in AD study, there's also PET scans, right? Amyloid et al., FDG. So how to integrate those together, right? So this is more like kind of like you look at this thing from different angle, right? So maybe some are related to more to this subtype than others. The amyloid might be the amyloid subtype, ATN, right? Tau could be kind of T, and in neurodegeneration, that could be the structural MRI, FDG. So I think this is more like from the horizontal perspective, right? You can actually look at each individual separately. You might be able to find a trajectory specific to that subtype. But if you put all those things together, integrate those together, what do you get? It could be something similar to the diagnosis, right? If you want to model the disease, the overall disease trajectory, you may need to integrate those things together, right? Because the diagnosis is a mixture of everything. So another thing we want to think about is not just these horizontal kind of integration, but also say maybe vertical, right? So we have data at different scales, at genetic level, molecular level, cellular level, and kind of this system level, brain imaging, fluid biomarker, and then cognition behavior, right? So if we want to, we can actually figure out the biological pathway from genetics to say molecular signature and brain kind of biomarker and to the cognitive outcome. So maybe there's some opportunities for say causal inference, right? So to figure out the relationship between that. So I think if you see multiple evidence at different scales, the signal is reassuring. So that can help you form like a very meaningful hypothesis for subsequent analysis. Right?
I think I'll focus on pipelines. They are really important, and we know that they matter a lot in terms of the final result. But I think unlike I don't think everyone can be hung to and redo it all themselves. So I think if you're going to be a statistician and you're going to focus on developing methodology, you must depend on basically using someone's prepared data and so that's I feel like to make a statistical contribution, you kind of have to do that. But if you think carefully about the impact of choices that come with pipelines, like Martin talked about yesterday, these issues about when you correct can be really important, and there's huge variation in that. So it kind of yeah makes me sad because it's really important, but in some ways you kind of have to set it aside if you're going to, like, you know, in any kind of monetized group, make an impact. I mean, I think about the you've probably heard of fMRI prep. fMRI prep was created because they said, people just mindlessly use SPM or just mindlessly use FSL or just AVNI. We're going to carefully pick and choose between all these different tools, find the best brain extraction, best motion correction, best. Great. Except now people just use fMRI prep mindlessly and assume that it's a good combination. And it probably is, but you know, things have changed, and so they're trying to improve it. So I think that there is a way, and if you're interested in actually studying this in itself, I think there are some interesting methodological options there. And I've worked with some people doing these multiverse analyses. So this is where you take one data set and you consider a whole spectrum of pipelines, and then you see what sticks out or what stays across them. And I think this is, if you really want to be able to trust a result, you want to see that result over all the different variations of your pipelines. It looks like a sensitive, yes, a specification curves, but and it's a fun. You can do that kind of sort of qualitatively, but I think it's a really fun statistical exercise because it's a great example of, I call it a same data meta-analysis, right? Because you start with the same data, you're going to analyze it like 20 different ways, and then you're going to get 20 different results, and now you want to put them together. Well, you can't do a meta-analysis because they're dependent, and it's not even like a typical hierarchical model because it's not like they're symmetric or exchangeable. Some of them are probably going to be very similar because the options are very similar. Some could be very different. So it's something that I've been working on, and but I think it's an interesting area where there'll be lots to do.
I have a question for you, Tom. So what about disease modification in the brain, right? So when we run all these pipelines, I mean, different pipelines. For example, you know, you want to run a segmentation SPM on an AD brain that has a lot of atrophy. And then we try to do a registration. Martin was talking yesterday how you're trying to pull the brain in all this direction and do these population average maps. It makes a lot of a difference what you're processing it with. And then, you know, you add motion and all of that. So there are pipelines, you know, depending on the pipeline, sometimes we struggle, you know, using free surfer and combining it with different things, which becomes not reproducible if we want to give it to.
It's super important. Yeah, I mean, I think of it for the UK Biobank, the initial plan was just like, well, we'll take FSL off the shelf and just shove it through that. But of course, FSL was developed for mostly younger, healthy people, and then suddenly just these average British older people with weird looking brains and monstrous ventricles suddenly, you know, kind of broke FSL. So they had to do all these kind of modifications to get it to work. And so, yeah, it's, I mean, it's super important, but it's hard for me to get in. I don't want to do it. It's super important.
Right, so-
it's
so
not,
in the-
it's engineering. So like, you know, I'll help my engineer friends do it, but it's
in PET imaging, they used to do standard manual segmentations when probably when you were at the Pittsburgh Center.
I've drawn
So
many
that was
regions.
exactly, we drew many regions when I was a graduate student. So you would manually trace those regions and then you would go register that with the PET scans. Not that was the best thing, but that was, we did a lot of inter-rater reliability studies. We did a lot of, you know, there are a lot of people that would do that manually. And we've done some of the reader studies tracing some stuff in the brain. So that way you would call that the gold standard, but there are several people that I talked with that they said that's not very good either, because you have a lot of variability from the person that is actually drawing the brain. So
I often sometimes say, like, you know, if Carl Friston had never been born, like maybe we only would ever analyze ROIs, and like we would never be doing VoxWise.
Can you comment at all about longitudinal imaging?
Well, you should come to my talk on Thursday and learn all about longitudinal imaging. But it's extra important, and it's all the more important because, of course, issues with the scanner change and harmonization. It's sticky. It's really sticky.
Okay, thank you. I just guess maybe I should put this panel section at the end of the day because all the topics should be involved before this panel discussion. Okay, let's see the next one. So yeah, we'll see that. So if we want to successfully process and integrate, also we want to do this data analysis for all these studies. it not only demands this kind of expertise in statistics called data science, it also needs expertise from other different disciplines. For example, neuroscience, epidemiology, and genetics. So we know for our individuals, it strongly depends on our research background. Maybe we can only handle one or two, but I just want to know that for the panelists here, how do you guys manage or balance, integrate these diverse skill sets?
That's a great question. So obviously you need a big, you need a team. It's great to work with experts in each of these fields. I think you should have like you don't have to have expertise in every one of those topics, but you should have some familiarity with what the current trends are and with like what some of the basic issues are, like especially in neuroimaging, genetics, and if you're like a statistician, epidemiology and statistics, obviously. I rely on working with really smart people who care about being multidisciplinary themselves, but have expertise in each of these areas. Yeah. But yeah, I mean, and who knows if we're getting the balance exactly right? Yeah.
Yeah, I totally agree. You cannot have all those skills, or maybe there are people that do, but you have to rely on the team of experts. So I work with a lot of people that do the processing, especially, PET and MRI, but you have to understand a little bit what is happening and how this is done. But I think you have to rely on a team and create good working relationships with your collaborators. So otherwise, it's not possible.
Yeah, I agree. Yeah, I don't know if Muchen is still here. Actually, he teaching me that I should hire kind of postdocs from different disciplines. Yeah, no problem. So I can actually learn a lot. Yeah, from my team member. Yeah, I do have a postdoc. Actually, he's he's an expert in like biochemistry and pharmacology. So since we study like a drug repurposing, so he can really teach me how to say if we have a target, then what's the druggability and how to do that, right? So actually I learned a lot. I think another thing is we should be open-minded, and we need to also learn some domain knowledge, although we are working in the middle, right? But it would be good to learn kind of both. And yeah, so another thing is regarding the strategy, how to do that. I think I kind of I think probably in all the universities there are a lot of like interdisciplinary kind of centers or institutes, right? So you can attend their like events or retreats, right? Talk to people. And another thing is to serve on the say thesis committee on like kind of students, right? Then since we're all working in this kind of interdisciplinary field, right? So you can actually learn a lot from other committee members, right? So, and maybe also co-mentoring with other kind of advisors. So in that case, I feel this is really, really important. I think probably the most important thing is to define the problem, right? We do need a domain expert to define the problem. So since actually, I mean, I'm not in biostatistics, I'm in biomedical informatics. So I really kind of dependent on the domain expert to define the problem. And I would rather to do like a problem-driven research. So kind of instead of I come up with some algorithm, then look for applications, right? So
Yeah.
it's very important.
I'd like to double down on what you just said.
Yeah, no, I guess what I would focus on is graduate student education, and you can't like be trained up in everything, but I think it's really important to pick up as training statistics PhD to if possible give them some formal training in we're here in neuroimaging, right? So I was very fortunate in Pittsburgh. They have the Center for the Neural Basis of Cognition, so that was like I got a brain in a bucket and like we had to do dissections. That was invaluable for me to like make me someone who, I'm a better collaborator because I can communicate with the people about neuroscience and cognitive neuroscience. And in Oxford, I'm very fortunate. We have a graduate, it's basically a 2-term graduate training program where I can just ship off my graduate students to learn about MRI and MR physics and a bunch of stuff that they never use, but then they actually now have that grounding. and MR and now MR acquisitions and basics of analysis. So it doesn't happen everywhere, and you may have to do it ad hoc, but I think it really provides a really great grounding that will enrich their subsequent research.
And I mean, these are all fascinating areas, like genetics. It's amazing how rapidly it's moving. And then like neuroimaging, obviously, we all think that that's fascinating. I think, and statistics, epidemiology. I mean, like, so like just express curiosity, and if you have someone like really amazing experts to talk to, just ask them a lot of questions, and that goes a long way, I think. So.
I think at the University of Pittsburgh, at the PET Center before COVID, we would have, so PET Center would actually provide graduate students from the biostatistics and would let them come to the PET Center, and they would be there, you know, with the physicists. They would see the scanners, they would see how data is processed. I thought that was great. That's how I kind of learned the imaging. So just being surrounded by people with different background, so at least have a basic understanding of what the steps were and what we were doing. So that is great. If you are a graduate student, if you're at the university that have these centers, you know, maybe it's worth trying to see if you can integrate with those groups and learn from them.
And several people have asked about how to get a seat at the table. If you talk to people and take the conversation seriously, you get a deeper understanding as to what concerns they really have, and like maybe they don't even know what concerns they have, but you can kind of glean it from the conversations with them, and that's valuable to you know, with your coming at that with your expertise. So.
I think you have to do a lot of volunteering to volunteer to be in different committees, in different centers, to do a lot of different.
Yeah, like Rob said, you know, if they wanted a t-test, help them with a t-test, but that's like the bridge to get to know them.
Okay, thank you. So I think let's move to the next topic. That's about the new technology. So the first question is, so as we know that, so for this emerging and generative AI technology, such as this, we know this the AlphaFold. So this is very famous because it's successful. They solve this modeling of protein structures and interactions. So we would like to see that. So for all these panelists, so how do you find that? So which area of the population neuroscience research will be the most exciting in terms of benefiting from these new technologies? So from both methodological or in the application parts.
So I think I'm fortunate that I'm getting close to retirement age, because I'm a Luddite, and I see the dangers more than every time I see people blindly apply AI methods to data sets that I work with. It always comes out with like basically garbage. So I'm skeptical, but what do I know? I'm probably like in 2 years it'll be
You're a
better.
dinosaur.
So and I care more about like kind of like getting understanding from the data as opposed to prediction accuracy, and then I think we come back to what Martin was saying yesterday about the dangers of blindly putting stuff into an algorithm that will just optimize prediction performance. So I'm sure that will be solved, and I'm sure that I'm, you know, like that, but it'll be solved after I'm retired, so I'm not going to comment.
Can I just say no comment? I think that, you know, it's we have to know about these sort of emerging techniques, and I have graduate students that know a lot more about these, and they're very proficient. But I think I'm old-fashioned. I always like to do an analysis based on hypothesis tests and try to tell my collaborators to do that. But unfortunately, I mean, it's, you know, unfortunately, we have these tools that we can use to handle large data sets. So I'm sure we will be in a better place in a couple of years, probably.
Yeah, so I feel actually these are kind of like a relatively new, like a new platform, like a new infrastructure, right? So we are actually using some of those in our analysis, basically, because the goal is like, it's more like the foundation model, large language model. It's all about encoder and a decoder, right? For the encoder, you want to learn an embedding, which is just the feature extraction, right? They just have a new name, right? They call that embedding, right? You just define new features, right? And And then, so the goal is we want to extract some meaningful features using those foundation model or large language model to help us to perform subsequent tasks, right? If you want to identify biomarkers, then basically you have to kind of separate the patients versus controls, right? Then identify relevant features. You want to find such a feature space, right? If you want to identify the subtypes, then you just put everything into a new embedding space so you can see distinct cluster structure, right? So if you want to figure out the trajectory, you want to kind of map those into an embedding space so that you can see a more like a linear trajectory, right? So say, ideally the healthy people are here, say the dementia patients are there, in the middle could be the MCI, right? So you can model the progression. So I think we can use those models. And another thing is about the decoder generated AI, right? You want to generate new things, right? So like these vision language model, you have the image, you have the radiologist to report, right? If you see an image, if you can use a model to generate that report, that can actually help a radiologist. You don't want to use that to replace the report, but the radiologist can read that, make the final decision. At least it provides some additional information, right? So in terms of neuroscience, I think there are a few directions. One is AlphaFold. Why don't we kind of integrate biology into neuroscience, right? Because we also need to find a target, right, in the brain. So that the protein in the brain, we need to find a compound that can bind the protein, then build the drug, identify the drug. So we can use that, right? And for virtual kind of for screening, right? And so there's also multimodal AI. So people are aligning the text, image, audio, video, tabular data, right? We have to have those, right, in neuroscience. So we have images, we have text, right? EHR, tabular or also text, right? And another thing is like for the structure, they actually use these for AlphaFold. They are inputting this structure, not just the sequence, but also the structure, right? We do a lot of like the imaging study, like we look at the images, the surfaces, the shape, right? So there's a point cloud, right? Network topology. These are all structure. We can actually make full use of this new technology, right? And other things is, I think yesterday we talked about these multimodal scale, right? So micro scale, meso scale, or like micro scale, then we have to have data. Some data are at the neuronal level, right? So we have single cell spatial transcriptomics, right, from the brain. So you can actually make use a lot of those tools, right? And yeah, these are just some examples. I feel probably we should catch up with all these new technologies. There's a lot of applications.
Yeah, I don't think I'm a dinosaur, but I don't have much optimism that they're gonna make a big impact for our field because things like AlphaFold and Go, they're working with these discrete things. There's like yes, no, is this a real? Does this protein fold like a real protein? Does it? And you get these up down. But when you think about the resolution by which we can image the human brain, in vivo, it's like we're just so millions of miles away from sort of getting hard answers to train an AI to do something. So I think it'll be useful uses in doing pre-processing. We're using generative models to help make training data for lesion segmentation when we have a shortage of kind of certain types of image data, whether it's like thick slices, like that actually could be useful. Though actually, the one application which is really intriguing, which is generative models for basically helping radiologists to have a fresh result from a colleague told me about, which is that he made this system which would produce text from an image and give that to a radiologist as a suggestion. So he did a trial where he compared that to also he can point to an area in the image, just like reading chest X-rays. And then he did this comparison, which one helped more. But he did something on like 30% of the guides, they're wrong. They gave them bad advice. And the radiologist easily found the bad advice on the image, like look over here, and they like, that's wrong thing. But they almost always took the wrong written advice that looks like a radiologist report. They're like, yeah, that sounds right. Yeah. So there's gonna be lots of issues of actually putting this to work and helping radiologists.
Yeah, I want to add something about this part. And actually, the large language models get extremely popular in the last several years. And since the ChatGPT, the ChatGPT is a good tools for something, but not for everything, right? And we know that. But you only understand why these kinds of, there's the two most important foundation models. One is for computer vision. Another one is for the large language models, NLP problems. The reason is because the tech company has a ton of money to annotate the data. And for all these possible downstream tasks, okay? You think about the ChatGPT, how much money the investor just annotated. I think maybe 80 or 90% of the cost on that aspect to me, okay? The building model part is not that difficult. Everything is computing powers, okay? After you have good data set, okay? And because for CV problems, also NLP problems, for many tasks, downstream tasks, pretty simple, okay? Because at least human annotators can easily identify the ground truth and use it, you know? That's the reason, like self-driving cars, all these things have been improved a lot in the last several years. That's the key reason behind it. And but for the neuroimaging, in general, neuroscience, there's three tasks that are important. One is the segmentation. Okay. The second thing is they can use the do ray tracing. Okay. There's a foundation model for segmentation. There's a foundation model for ray tracing. Many people jump inside, just do it. Okay. And also there's like Yushin said that you can use the data to do the imputation. Okay, the foundation model for data imputation. We do a lot of missing data problems. You can use the imaging to predict the text and give the radiologist to do validation and do these kind of things. If you accumulate enough number of cases and do annotation, at the end of the day, I think that will save a lot of time for the radiologist. And I think the current days, the barrier is all goes back to the data set itself. Whether you could get good annotated data set for all the important downstream tasks. If you can't, answer is no.
You won't ever. I mean, when you consider all the different things that radiologists see, or I'm not sure what your use case is exactly, but
that depends. That depends. You know, I think this is, if you think about it, there's a foundation model for segmentation. There's the CVPR has this year have a dream challenge. Is basically doing multi-model, multi-organ segmentation. And that's basically some company provide a baseline models, and basically all the teams jump inside to take over that basically foundation model they trained at the beginning, and then modified, add all kinds of tricks inside to try to improve that model. I think that people try to push on that directions. I think that just like Tom said, that it could be used for people processing the data. I think at the end of the day, one big issues in neuroscience in general, we don't really have a good data set, annotated data set to validate all these segmentation ray tracing algorithms in different come from the different studies, you know, and also different scans and all the things. But we do need that at the end of the day, maybe next 5 or 10 years.
Yeah, I agree. I think it actually depends on your problem, the task, right? So now at least we have a choice. So you build this foundation model that can kind of deal with most of case kind of pretty satisfactory. But if you have your own data, so one way is you can actually use the foundation model kind of to try to fine tune that, right, using your own data. So hopefully it can kind of generate a better result than apply that directly, right? Or another thing is if your problem, so the problem might be simpler than that. If the problem is simpler, why don't you just train it by yourself, right? There's no need to use foundation model. So a simple model might work better, right?
I
So
think
that's-
for most of the cases, simple, linear models works, right?
Yeah,
In
yeah.
many cases, we do think of the traditional statistics still valid.
Right, right. It just, we have a different choice, right? We have more choice. You should try both, I think. Yeah.
Okay. Yeah.
I know is ChatGPT is really good for writing letters of recommendation.
And also can help us to find the definition of population neuroscience.
That was actually a pretty good definition.
Yeah. Okay, I think the last question for this discussion is, so we know most of us teach the courses on statistics and data science and medical imaging processing for both graduate and undergraduate students. So now we have all these new technologies. So do we still need to emphasize these fundamental techniques in your teaching, or we can try to switch to these new technologies?
I don't teach, so I'm going to pass.
I don't teach, I do small groups of consulting, but I think that we should emphasize the fundamental techniques and probably mention the new ones. You always have to add the new stuff so the students would know about it, give them information where to go and look and, references. I don't know if we should just entirely switch to these new technologies.
Maybe not teach, just training the students. Yeah, you need to give them some suggestions, and if you encourage them to try to
Exactly.
reach out to these new technologies.
Yeah, actually, I do teach. So I teach machine learning and health application. So basically, my strategy is like 50% versus 50%. So half of my courses are lectures. So I teach these fundamental concepts and models. And the other half are more like, since my title is a health application in it. So we just kind of do a literature study. I think this spring I asked a student to just read the Neurex paper last year, right? And select a few interesting ones, and I assemble all those together, and we did a topical modeling on all these the abstracts, right? And then we have all these topics. I think around 10 topics, and I group all those students based on their preference, right? Then we have these 5 different sessions. We pick the top 5. Then they can present what one of the papers they selected. So in that case, we can learn kind of state of the art, cutting edge, new development in machine learning. And what they select, since I'm teaching biomedical informatics or related biology and kind of medicine, right? So a lot of those are about like just AI in health or drug repurposing or computational biology. So we actually learn both the methods and also applications. I feel this is actually pretty effective, right? So I feel in terms of we do need to emphasize these fundamental technology, because the key thing is even you have this new platform, a lot of new development are kind of, a lot of those are just based on the same intuition, right? So say, if you analyze these, say, the sequence, right? NLP, previously you use, say, recurrent neural network, LSTM, right? And GRU, and then you find out actually it's very time consuming because it reads things sequentially, right? Very long sequence, it takes a lot of time. Then people develop a transformer, right? The transformer is only look at the segment and regard to the context, and you can parallelize that. It will become more efficient, right? But with the data getting bigger and bigger, transformer becomes very expensive, right? If you do the computation. Then people go back to the original model and look at autoregressive. You only look at a small segment, right? The Mamba, so these new infrastructure, it's more efficient. Now they want to kind of accelerate the transformer now. But all these concepts are kind of the existing concepts, right? So we need to understand these basic concepts. So that's why I teach. The textbook I used was kind of written, I don't know, maybe 15 years ago. I think it's still not outdated. So all these kind of concepts still applies, right? Previously, I teach students like a model complexity, right? So why kind of typically you want to look for simplest model, right? Then it will be easier to generalize if it works, right? If it's not working, then you have to try more complicated ones, right? But now, if you have these deep neural network, you have thousands or millions of parameters. Based on the traditional methods, actually, it shouldn't work, right? Because it's very easy to overfit the data. So now we actually we need to change, how should I say that, the catch up, right? People are studying like a data complexity. The thing is all these images are lives in a very low dimensional space or in a manifold, right? So that's why all these kind of complicated neural network can learn this manifold, right? So I think we we need to update that a little bit, right? But the fundamental concepts are still very important. Yeah.
I don't think it's a manifold.
Oh yeah, man. Just an example. It may not be manifold, right? So it could be a graph or other relation, similarity learning, right? Well,
I want to take your class. That sounds great. But yeah, so I don't have much to add. Yeah, you need both. You need the foundation. Just another way to say it is that when the sort of the fancy, complicated methods might go off the rails, you need to know enough to know what, how does the simple method compare, right? And you should be able to understand why you're getting different answers from different methods. And if you only know the fancy ones, you won't know, how the basic methods work.
Okay.
I think one thing is actually we discussed in the statistical community about how do we reform the current curriculum, in what sense. Actually, we organized multiple events to discuss that. Actually, we spent 3 days in Munich, Boston, and Peter basically organized these events. Actually, we have the concrete suggestions how to modify the current undergraduate and graduate training programs, and how to condense some of the existing courses, like similar courses, like multiple regression models. Do we need to really teach it as 4 courses, okay, from different longitudinal survival or linear model, whatever it is, you cover almost 2 years, is necessary or not? And then how much we need to want to add the existing machine learning models and to the existing training in what sense, and also the different major data types, because these days, most of traditional statistical training was focused on structured data. And even, Rob said that, statistics become too abstract in certain degree. Random variables is right in the functional space, but that in reality, that doesn't look like that, okay? And that's basically, we need like CMU has this 1 year basically program, basically for all the PhD students, they have to do a larger kind of data analysis project. I think that how much you adopt these kinds of good ideas to emphasize the students in statistics, all these biostatistics department, basically train them to able to handle, to touch the data, okay? And handle it, do it right away, process them, and doing the right analysis, good reports. That's something skills we need these days. It's not just because we have too many different data type, right? But you're not just I never touched this data set, so that I cannot do anything. Then the opportunity will give back to other people. Okay, that's the most important things. Nobody was trained to do many data types, right? You think about how many data types we touched in our graduate studies. Answer is no, right? Almost a little. But we still need to develop our students to handle new data type, know how to analyze, think about all the questions statistically, using the right statistical thinking, develop the method, and then write reports on this. That's important skills. We're not just prove another bond, okay? That's important for all the field.
Yeah, I agree. I think we should start from some concrete problem, right? So if you're giving student a problem, so they will figure out a strategy to solve that, right? So they can try simplest model. If it works, that's the best case, right? So if it's a data and it's very easy to interpret that, but in most of the case, it won't work because easy problem, probably you can think of a strategy, people already found that, right? So a lot of real problem, you need to think about new strategies to solve that, right?
So, I mean, but there are some problems that the simple approach works almost as well as the most complicated approach, and it's just a lot easier to apply. Polygenic risk scores in genetics. It's simple. It's like mass univariate analysis, and you do some pruning and whatever, and then you come up with a score. And people try to apply machine learning and all sorts of things to it, and it never does any better demonstrably than the simplest model. And so I think training on knowing when to use simple models and when it's likely to be valuable to try to apply more complex models, that I think would be useful.
Absolutely, yeah. I think yes, the first thing you should try simple model. So that's my priority, right? Yeah.
My strategy in last 5 years is I ask my student to do clinical papers, data analysis papers. I just say, this is the data analysis I want to do. But you just do it and write the paper up. We submit it. Okay, that's the basic how do I train my student to do that. Instead of before you do any method development, because I wanted them to analyze some data set. This is all these standard method that people are using. You just try to understand the whole pipeline from beginning to end. You write the clinical questions, you write it down and do analysis, write the reports, go through everything and tell me, what do you find anything interesting? Can we do something on that? That's the wild strategy I take in the last 5 years.
Okay, I think we are run out of time. So thank you today. So thank you for all these panelists, and also thank you for all you guys' attention.